So I just want to start by introducing you to some overall numbers of the microbiome. And I do want to preface this with these are debatable and these numbers seem to change every couple of months. However, what we do know is that there are certainly more microbes in our body than human cells. And there is certainly many more microbial genes in our body. So there's an estimate of there's 100 to 1,000-fold more microbial genes than there are human genes in one's body. I like to give the anecdote that a normal adult has 5 to 10 pounds of bacteria in their body. So the microbiome is extremely important, and importantly, it has been associated with disease repeatedly. So the statistic here says that up to 90% of diseases have been associated with some sort of alteration to the microbiome. So when there's an alteration to the microbiome, we call it microbial dysbiosis, and this is when a change occurs, and this can be a change in the actual bacteria or viruses or microbes that are present. Microbial dysbiosis can also be a change in what sort of metabolism or fermentation products they're making. But when this dysbiosis occurs, it has been associated with disease. And this is just a very short list of some diseases that I think are very relevant and important. And I also want to use this figure to point out the fact that we have microbiomes all over our body. So we typically think of the GI tract microbiome. And I'm going to speak mostly today about the gastrointestinal tract. And the GI tract is certainly where the majority of microbes are. So probably 99% of the microbiome in the body is in the colon. However, you have a skin microbiome, an oral microbiome, lung microbiome, a vaginal microbiome, which I will be speaking about at the end for a little bit. So there's several microbiomes across the body, and this is not all of them. Basically, anywhere you have a mucosal surface, when you have an exterior surface, such as the ears, there is a microbiome. So I think this is a good picture demonstrating that it's very important that we have homeostasis in our microbiome and we don't allow it to get altered. And this is just a good depiction using a yard or a lawn as an example here, where you have a flourishing gut ecosystem, where you have all of your microbes and your microbial products that are working with your body, and we're going to talk about how they work with your body in a moment. But they work with your body to maintain a flourishing ecosystem and promote health. If you give something like antibiotics, or you have a transplant and have to do irradiation, you can really devastate this ecosystem. Although, as I'm going to show you, we didn't really understand exactly the details of how the antibiotics would affect this ecosystem, so we really dove into that more for the study I'm about to present. And then after antibiotics, you can have, this ecosystem go wild. So the example here is that you have weeds growing out and you lose that nice lawn, depending on what the circumstances are. And this is when you're going to have things like C. difficile infection, vancomycin-resistant enterococcus, diseases that will take over and become very problematic in the GI tract. So there's several different ways that we attempt to alter the GI tract. I'm not going to get into this as much today, but it's things we're thinking about, and I will just mention that none of them are great. But you have things like probiotics, where you can give beneficial bacteria, such as lactobacillus or bifidobacterium. There's things like prebiotics, so prebiotics are food for the probiotics, and the prebiotics can promote good bacteria to grow out. And then there's things like bacteriotherapy, which would be a fecal transplant or full transplantation of a bacteria ecosystem from one host to another. And we hope that by altering this, then you get back to this good ecosystem and the good bacterial communities. But we really needed to better understand this. And so going back up to, this devastation by antibiotics, we really wanted to understand exactly what different antibiotics do. And this is very important. So the GI biogeography, this is very important why the bacteria are interacting with the whole body. Because it's not just that they're in the lumen of the intestine and not important. We wouldn't have 100 trillion bacteria if they weren't important. But you have the bacteria in the lumen, just to remind you here, and then there's going to be also be bacteria in the mucosal layer. And then in the lamina propria, you have a lot of beneficial immune cells that are going to play a role both in protection from pathogens, as well as homeostasis to maintain that epithelial barrier and health in the GI tract. And there's a lot of interaction and crosstalk between the microbiome and the immune system here. This is very important. This is very important as well because you have this anaerobic food web where the bacteria are extremely important in metabolizing food products and anything else that can come into the body. And you have several different classes of bacteria that can make these different, and I'm sorry, the lines got weird on this in the transfer. But there's several different classes of bacteria that are very important for making byproducts such as short-chain fatty acids, which are extremely important in maintaining epithelial barrier health and maintaining that immune system. And so the interactions between these microbes and how they actually have fermentation of the products that we put into our body, such as food, and then they have their byproducts such as short-chain fatty acids, and that interaction with the immune system is extremely important in promoting overall health. So one of the ways that this interacts, like I said, is the short-chain fatty acid, but I also want to introduce you to a few cells that are particularly important in the GI tract in the context of the microbiome as well. So we have dendritic cells that I won't talk about as much today, but that these are going to be our main antigen-presenting cell in the GI tract that are going to sample pathogens, and then they can interact with other cells, and antigen-presenting cells have also been shown to be highly affected by what microbes exist. And then you have things like Th17 and Th22s, which I'm going to talk about more in a moment, but that is a CD4 T cell that produces IL-17 or a CD4 T cell that produces IL-22. These are very important cells for mucosal homeostasis. And then you have down here these neutrophils, which are the first responders. And we'll get into that more here. So neutrophils are extremely important with bacterial infections and responding to bacteria in general. They have many antimicrobial functions. Neutrophils are, like I said, the first responder to an infection and interact very closely with bacteria. They can do this by phagocytosis, degranulation, or neutrophil extracellular traps. They also make a lot of byproducts, such like antimicrobial peptides, and those antimicrobial peptides can then regulate bacterial communities. They also make proteases, which can cause damage to the tight epithelial barrier if they're left alone, left there too long, and then reactive oxygen species. So these are all used to kill the pathogen, but I also always remind that it's important that we have antimicrobial functions to control bacterial infections, but we also don't want neutrophils around too often or too long because they do have these tissue damaging properties by making things like these reactive oxygen species and the proteases. I had mentioned these Th17 and Th22 cells. So these are cells that we have repeatedly found to be extremely important in mucosal health. So IL-17 and IL-22 can both promote the epithelial barrier health, and so if you have damage to the barrier, IL-17 and IL-22 can actually repair it directly. This is also important because neutrophils, the second line of defense is neutrophils actually recruit both IL-17 and IL-22 producing cells. So there's also this network between the neutrophils and these Th17 and Th22 cells. Th17s are often known for inflammatory conditions. They increase in things like inflammatory bowel disease and associated disease. I do want to mention, though, that this is an association, and actually if you give anti-IL-17, it exacerbates disease. So it's unclear still if the cells are there to come in and in response to damage and in response to damage to the epithelial barrier and alterations to microbial communities, or if they're actually causing damage themselves. But very important in mucosal homeostasis. So I'm going to jump right into our studies in antibiotics by kind of going back in time to a study we did kind of accidentally. So we do a lot of work in my lab using the rhesus macaque. It's a non-human primate model for HIV infection, and we can infect these animals with SIV, and it's a very good model for human HIV. And one of the things that we realized was happening is that when we were using this model for a vaccine study or a pathogenesis study, whatever it was, we found out that during the study, the vets were giving the animals routine antibiotics. And this was any time we took a lymph node biopsy or did some sort of biopsy in the animals were getting antibiotics. And so we stopped and said, wait a minute. So in the middle of our study that we're trying to understand how mucosal homeostasis is affected in HIV, these animals are repeatedly giving antibiotics. So we wanted to understand better what's going on when you give the animals antibiotics. And so this is just a snapshot of a small study that, like I said, it was kind of an accidental post hoc analysis that we went back to animals that had been SIV infected. So again, this is our model for HIV. And we looked at the microbiome of the colon mucosa and the stool. And so just to orient you here, and I'm sorry, the labels fell off on this one, but to orient you, each bar is one animal, and then each different color is a different bacteria. But what you can see is that pre-antibiotics, and this is the colon mucosa, so this is an actual colon biopsy, you actually have a predominance, this is Helicobacter here, so these are infected animals, the microbiome does not look good. And then you have a lot of changes in most of the animals post-antibiotics, and some of them did not respond at all. And I should mention that this was a Baytril, so interfloxacin, which is a veterinary antibiotic, which I'll speak more on later, and vancomycin that these animals were given. When you look at the stool antibiotic use, again, pre-antibiotics, you have diversity across that stool microbiome when there's many changes in all the animals after antibiotics. So we're seeing huge disruptions to the microbiome post-antibiotics. And another important part of the study is that when we compared stool microbiome, the colon microbiome, or a rectal swab. So you actually take the colon biopsy, so this is and then we take that tissue and it's snap frozen, so we're really looking more at the mucosa-associated microbiome. You have stool, which is going to represent everything that has gone through, of course, the GI system. And then the rectal swab goes around. What we were really interested to see is that where the colon, and if you're not familiar with PC plot, so this is a PC plot, and basically what we're doing is you take all of the data and put it in, and this is clustering the data based on certain attributes, and when you put it in, you don't know where it's going to cluster. And so this is really important because what we're seeing here is that these weren't clustering based on a time point or some other factor from the animal, but what they're really clustering on is body site, where the colon mostly clusters up here, the stool down here, and the rectal swab is kind of all in between. And so we think that because that swab is getting some of that epithelial barrier, as well as some of the feces, that it's really representing kind of an in-between. And I just wanted to point that out. That was a kind of a side effect of seeing the study that the rectal swab was good. But back to the data on the antibiotics, what we also see is, so we use a Shannon Diversity Index to look at how diverse the microbiome is. In general, when we look at the intestinal microbiome, higher diversity tends to be better. And what we saw here is that when you look at these different sites, in the colon, pre to post, you have massive increases in diversity after antibiotics. When we look at the stool, there's a significant decrease. And when we looked at the rectal swab, there wasn't actually huge changes in the diversity. So very interesting. And again, this is an infected animal, so they already have dysbiosis of their microbiome, and they're not having normal microbiomes. So we really wanted to go on to understand, and this was, like I said, just kind of a retrospective study where these animals had gotten antibiotics, and we thought, you know, how much is it disrupting the GI tract actually to give these antibiotics? But we next wanted to go on and understand what's the role in healthy animals, and because we want to understand when we give somebody antibiotics, what is it doing? And the reason we use macaques for this is because we can intensively sample. So this is the study design we used where, like I mentioned, we intensively sampled these where we have 4 groups of animals, 3 animals per group. It was a smaller study. And I'll talk more about each antibiotic in a moment. But throughout time, prior to, during, and after antibiotics, we got daily stool samples so we could really closely monitor the microbiome. We got invasive sampling, such as colon and vaginal biopsies throughout the study so we could understand what is going on in the immune system and actually look by flow cytometry, and then blood throughout as well. To give you a better idea of these groups of antibiotics and why we use them. So we did 4 different groups of antibiotics, cephalexin, or better known as Keflex. This is a cell wall inhibitor, broad spectrum antibiotic that is known to be partly effective against anaerobes, is what it is advertised as. Commonly used for respiratory infections, used for prophylaxis prior to infection sometimes before culture results get back in, and is used for multiple uses across infectious diseases quite commonly. You have clindamycin or creosin, which is a protein synthesis inhibitor, and also a broad spectrum antibiotic. It's used for several not as intensive diseases often, so things like acne, strep throat, second round bacterial vaginosis treatment, and we're going to look at these data relative to the vagina in a little bit. And Then we have the enrofloxacin or Baytril. You're probably not familiar with this one because it's actually a veterinary antibiotic. Because we were doing these in animals and because of the importance of the macaque model for things like Zika infection and HIV infection models, this is the most common antibiotic that is given to animals, and it's a nucleic acid inhibitor, broad-spectrum antibiotic that is told to be ineffective against anaerobes. And like I said, it's used quite frequently in veterinary practice. It's used when animals are moved between facilities. It's used prophylactically when there's procedures done. It is very commonly used, which is why we included this, because we thought it was very important for these animal studies. We also looked at paramycin, and this was an interesting one that we included. So paramycin is also a protein synthesis inhibitor, known to be effective against gram-negative and partly effective against anaerobes. Commonly used in HIV-infected individuals for cryptosporidium infection, which we don't see as much anymore, but particularly back before high antiretroviral use, we saw this quite often, and this was used often in HIV-infected individuals. Also used frequently in pregnant women. And the reason for this, and why this was an interesting antibiotic to include in this group, is because it has very low absorption rates. So paramycin is actually not supposed to cross the GI tract and get into circulation, and it's really only supposed to be focused into that GI tract, and it can also be used IV then. And so we wanted to include this one because when we look at the immune system and the downstream effects of antibiotics past the microbes, it was an interesting one that if you can change the microbes without actual absorption of the drug, are there differences between these groups? So the major methods that I'm going to talk about today, so these were all female rhesus macaques that we used. Like I said, there was 3 per antibiotic group. We took the tissue biopsies, blood or stool. The tissue biopsies were processed immediately, and we used flow cytometry to look at immune cells in the different tissues. We took snap frozen biopsies as well as stool and processed for DNA extraction that we did 16S rRNA sequencing on for the microbiome. We also took the stool and we did gas chromatography mass spectrometry for the short-chain fatty acids to identify what short-chain fatty acids are there. And then what I realized is not on here that I will show is we also took the plasma and looked at microbial translocation. I'll be showing soluble CD14 later. And then, of course, data analysis and lots of hard work by my staff. So jumping right in. So again, I'm going to just orient you to these plots where what I'm showing here, these are going to be a combination of the 3 animals. So it's going to be the average bacteria from all 3 animals per group, and this is across study day. So you have the first 2 baseline time points, day 3, 14, 28, 63 post-antibiotics. These are grouped by the different antibiotics, where you have enrofloxacin, cephalexin, paramycin, or clindamycin. And then again, each color represents a different bacteria, which is labeled over here. So what you can very quickly see is that very rapidly after antibiotic treatment, you see very distinct changes, very, very major changes in the GI tract. And this happens very, very quickly, and then it reverts very, very quickly too. And in virtually every animal, by day 28 to 63, you had a complete normalcy of that microbiome. And again, these are healthy animals. So I'm not interpreting this at all about how this is if we're trying to treat a specific infection, but it is very interesting that we see very similar changes despite all of these very, very different antibiotics, and you see it very rapidly, and it comes back quite rapidly, which kind of tells us how much our microbiome is difficult to really change, which anyone who's tried to treat an ongoing infection probably already realizes this. And I will get into some of the specific alterations in a moment. So when we look at the stool, again, the stool microbiome looks very different than the colon microbiome. What I just showed you is from colon biopsies, so those are going to be more of the mucosal adherent bacteria. But when we look at the stool microbiome, again, there's more diversity overall starting out. But as we go through antibiotic treatment, you can see that there are large changes when we look across the treatment, but that it typically reverts back to normal by day 49 to 63. This again is just looking at these cluster plots, the PC plot, where we're looking across days throughout all of the samples in either the colon mucosa or the stool. And what you can really see is that where you see the biggest clustering is this very early during the treatment. So day 0 is actually the first day of treatment, so they've gotten hit with the antibiotic, and then we take the sample, so they're already seeing the antibiotic. It works very quickly. But what you can see is that red, that 037, very, very rapidly after the antibiotics is where you're seeing that there's this major shift. But like I said, I wanted to go into some of the very specific microbiota that were actually altered, because what was very interesting is that there was significantly altered stool bacteria that tended to be actually pathogenic species that came out during antibiotic treatment, and almost seemed to be a bad thing after antibiotic treatment. So this is just a relative abundance, and this is all of the groups together now, so that all the different antibiotics together. Where in green you have the before antibiotic treatment. Red was the during antibiotic treatment, I believe this was day 3, and then after was the day 63. And so you can see these are several bacteria that are very interesting and have been associated with infections and disease. So Escherichia, which of course E. coli, we all know, Enterobacter, which has been associated with disease, Proteus, which is a highly pathogenic and highly inflammatory bacteria class. So Streptococcus, which tended to continue to increase actually. Akkermansia, which we had increased in all of these, and there's just discussing this bacteria this morning, so there's some interesting data coming out right now that we're trying to better understand the role of Akkermansia in disease that has been associated with IBD inversely. And then Ruminococci, which is actually a very beneficial bacteria that is decreased after antibiotic treatment. So you see these very specific changes in these bacteria that have been associated with diseases, which does give some cause for concern. Do we really want to be altering the bacteria? And cause for concern because, of course, antibiotics are extremely important and we need them, but when they're given so loosely for different things that maybe we don't need them for, do we really want to be disrupting these bacterial communities like that with, potentially pathogenic bacteria? So I talked about short-chain fatty acids earlier. We next wanted to look at, so we know that there's this disruption in the microbial communities, and what does that mean for short-chain fatty acids? And again, these short-chain fatty acids are fermentative byproducts of bacterial metabolism. There's the biggest class of bacteria that do this are the Bacteroides, and butyrate's probably the most famous of the short-chain fatty acids. Butyrate is well known for maintaining the epithelial barrier homeostasis. Butyrates can also promote different alterations to immunity that seem to be beneficial. Loss of butyrate has been associated with many diseases like inflammatory bowel disease and cancers. And right now there's a lot of studies looking at butyrate treatment and how that might actually lessen disease severity. But there are several short-chain fatty acids, and we looked at acetate, the butyrate, propionate, and valerate. And what you can see is of these short-chain fatty acids, all of them are rapidly decreased during antibiotic treatment. So the gray bar there is showing you the period of antibiotic treatment. And for every single class of antibiotics, you have this rapid decline of the short-chain fatty acids, which is interesting because the specific bacteria that are known for making the short-chain fatty acids aren't necessarily hugely impacted when you look at that by sequencing, but these data really make us think that possibly antibiotics are not just killing or getting rid of certain bacteria, but actually altering the metabolism capabilities or the actual function of the bacteria. We next went on to look at the immunology and what's happening in the lamina propria of the colon during this. So I showed you neutrophils early on, and we're very interested in neutrophils because, again, they're very responsive to bacteria, very important for protection against pathogens, but also can cause a lot of tissue damage. And so it's, there is this fine line that we have to regulate, and what is very interesting is that very rapidly, again, during this antibiotic treatment, and coinciding with when we have the alterations to the short-chain fatty acids, alterations to the bacterial communities, you see this rapid rise in all the groups, including the paramycin, which isn't supposed to be absorbed. So we do think this is an effect of these bacterial communities changing, where you have the neutrophils rapidly increasing in the gut, but then of course by day 63, they're all back down to normal. Oh, and sorry, that was a weird animation. That's just showing the flow cytometry and how we're measuring our neutrophils. We went on, we sent these samples to Mark Cameron's group at Case Western Reserve University, and they did RNA-seq so we could understand what's happening in gene expression pathways here. And confirming the flow data, what we saw was that genes like CXCL8 and IL-8, the IL-8 production pathway, which are both extremely important pathways in neutrophil recruitment, and these were raised in all of the different treatment groups as well. And it was the CXCL8 and particular is a little more variable, but throughout time what you can see is that the antibiotics certainly induce an increase in these neutrophil-associated pathways, even when we look at a gene expression level. So that was a nice validation of that flow cytometry data as well. Like I said, the neutrophils are really important in that barrier damage and interacting with the barrier, and the neutrophils can directly cause damage to the barrier. So we also hypothesized that alterations to these microbial communities that we see during antibiotic treatment could induce microbial translocation systemically. And so if you're not familiar with the concept, microbial translocation is where a microbe or a bacteria or a bacteria product actually translocates and gets across that epithelial barrier and gets into systemic circulation. And we're lucky enough to have Dr. Danny Dewick in the audience, who was the first one to show that in HIV infection, for example, microbial translocation is highly associated with disease progression and drives chronic inflammation. So you don't want microbial translocation, which I think we can all assume no one wants bacteria floating around their bloodstream. But what you can see here is that quickly after antibiotic treatment, during treatment again, you have this increase in microbial translocation. So, and again, and we see this with the paramycin as well. So, it's very interesting that it's not just you give antibiotics and there's no changes, or you just change the one bacteria you're targeting. You're changing multiple bacteria communities, and this is now having effects all the way into your systemic circulation and potentially causing systemic inflammation. Finally, I mentioned Th17s and Th22s before. Again, reminding you these are CD4 T cells that produce either IL-17 or IL-22, have been consistently associated with mucosal homeostasis and disease, and very interestingly are recruited by neutrophils. And you can see that, and this is now interesting because these cells, which again are part of the adaptive immune system, they're not going to be your first line defense like a neutrophil, but after antibiotic treatment, you see an increase in these cell types. And so what we really think these data indicate is that those neutrophils, the bacterial communities are changing, loss of short-chain fatty acids, it's saying something's happening, bring the neutrophils in. Those neutrophils are coming in, can cause barrier damage, and then the neutrophils are now recruiting the IL-17 or Th17 cells as well. And those cells we think are coming back in to really repair that barrier and say, okay, let's get things back right to normal. And again, this is all conjecture and correlation, but this is how the timeline of events seems to be working out here. And then again, confirming, and sorry that looks a little fuzzy, but we again did this RNA-seq analysis, and what you can see is there's massive rises in the entire IL-17 pathway after the antibiotic treatment. So again, this isn't during, but this is kind of the response after. And so this whole pathway is increasing in our colon biopsies. So again, we believe that after all of this is happening, that it's trying to trigger the Th17 cells to come back and fix things. So just to kind of summarize this part of the talk and what we see is going on before antibiotics, you have these healthy, stable microbial communities, hopefully, with your short-chain fatty acids that are promoting epithelial health and also having good interactions and homeostasis with our immune cells, where you have your homeostatic IL-17 and IL-22s and neutrophils, but at low levels because you don't want excessive inflammation. And then after antibiotics, you have this altered microbiome that tends to consist of more pathogenic bacteria. During treatment, you have lots of short-chain fatty acids, potentially damage to the barrier that then is increasing microbial translocation systemically, and you have this huge increase in inflammatory cells such as neutrophils, followed by an influx of the Th17 and Th22 cells. So hopefully I've showed you here that there's minor disruptions, the colonic mucosal immunity, which returned to normal by day 63, no matter what group of antibiotics it was. The disruption of the colonic mucosal homeostasis was demonstrated by increased neutrophils, IL-17 and IL-22 producing cells, and can potentially contribute to increased colonic immune activation and target cell accumulation. And so this has a lot of questions that it brings up about what does this mean for disease. And specifically because I work on HIV a lot, I think a lot about when we're giving antibiotics to, say, someone who has HIV, and we could be really disrupting the inflammation and immune activation that's going on that's so important in the GI tract. And again, just pointing out that it's really important to not just give antibiotics for anything, because you are causing very large changes. Even in a healthy system, there's huge changes that occur, tend to go back to normal, but could exacerbate disease if they are unnecessary. And again, I am not hating on antibiotics. I think they are a wonderful, wonderful thing, but again, just understanding that there are consequences in the immune system as well as peripheral circulation. So I'm next going to switch gears for a little bit for the rest of my talk, because I think something that came out of these data that's really important is understanding how oral antibiotics might affect the female reproductive tract. So the female reproductive tract microbiome is extremely important and is associated with several diseases. So we typically call it bacterial vaginosis when the microbiome is altered in the female reproductive tract or in the vagina. And it's consistently shown that if you have bacterial vaginosis or a dysbiotic microbiome in the vagina, that this is associated with things like HIV infection, preterm birth, several STIs like HSV, increased yeast infections. Overall antiviral immunity is altered when you have an altered microbiome in the vagina. So we asked the question, how do oral antibiotics affect the female reproductive tract? Because this is something that is very, very understudied. The female reproductive tract in general is understudied, but in terms of how oral antibiotics might possibly affect this, has not been well studied, especially in terms of immunity. And so we had a nice opportunity here, given that we had these actual vaginal biopsies throughout the study, to look at this. So I'm going to do this backwards now, and I'm going to start first with the immunology. And so, again, we have our 4 different treatment groups here. And so, again, these are the same animals I just showed you, but now we're looking at what happened in the vagina after treatment. And when we look at the vaginal CD4 T cells, and the reason we looked at just CD4 T cells in bulk here is because CD4 T cells are the major target for HIV. So when we think about giving a healthy woman an antibiotic, and if it has an alteration of CD4 T cells, it could have impacts on increased HIV transmission. And sure enough, we do see that after antibiotic treatment, you see this increase in CD4 T cells in the vagina, which was very interesting. And it was a bit different between groups, but you overall saw all the groups increase. I also looked at CD8 T cells to understand if we're having kind of an immune response here and what's going on in overall driving adaptive immune response in the vagina after this. And this is staggered across antibiotic groups, very different, but overall tend to see increases. And very interesting with clindamycin, that was an increase that went up and stayed up. You'll kind of notice that the clindamycin and the cephalexin actually tend to have the biggest alterations. Well, and I'll come back to these data again, but they tend to have the biggest alterations in immunity, and when we look at the microbiome, we're going to see some evidence that might support that from the microbe standpoint as well. I told you about neutrophils in the GI tract. Neutrophils are also extremely important in the vagina. Actually, in the lumen of the vagina, you find the majority of the immune cells are neutrophils, actually. And neutrophils are associated with damage to the epithelial barrier that protects that female reproductive tract as well. So again, the neutrophils play a very important role, not just in pathogen protection, but also have this dichotomous role where they can actually induce damage as well. And we do see a spike in vaginal neutrophils, particularly in the paramycin and the cephalexin group. So again, just to bring it back and just show all these data together, these are the same figures, but what I wanted to point out by showing this all together is when you look at the purple of the cephalexin or the red of the clindamycin are where we really tend to be seeing the biggest alterations in the vagina. And again, remember the clindamycin is typically used as a second-line bacterial vaginosis treatment. Metronidazole is the first antibiotic that's usually given for bacterial vaginosis, followed by clindamycin, or clindamycin is used instead of metronidazole at some point. So this is an antibiotic that's actually given for the purpose of treating bacterial vaginosis. So here we're looking at longitudinal taxonomy of the bacteria changes by animal. And again, the vaginal microbiome is much different in a monkey than a human, so I will give this caveat, is that monkeys, of course, do not follow the same hygiene rules as women typically do. And so there are large differences in the microbiome, but I think that it's important to kind of look at the overall how it can affect community structure. So here I have each animal grouped out. So each kind of cluster is one animal, and then its time points throughout the study phase, and then again grouped by the antibiotics. And I know this is kind of a lot of data overwhelming to look at, and just showing you kind of overall that you start seeing changes, and some animals have more changes than other animals in the vaginal microbiota after the treatment. I think it's a little easier to look at it like this, where now we're looking at overall phylum level changes when we started by regimen, and so now we're looking at each regimen and the average of the animals. And where you see these changes in the lines is where we're seeing these alterations in the microbiota, and the red line indicates where the antibiotic treatment started. And again, similar to what we saw with the actual immunology in the vagina, when we look at the microbial communities here, the cephalexin and the clindamycin are where we're really seeing, not huge changes, but we are seeing significant changes. So when we actually take this out and look at the cephalexin across time points, or within that actual group, what's very interesting is that there's potentially pathogenic genera that were increasing very early again after the cephalexin treatment, things like Prevotella, again a highly inflammatory bacteria that can cause barrier damage, Catinobacterium, Enterococcus, and actually Shigella was increased, highly increased in these animals. So, and this, on the left here, it's just showing what the full changes throughout the treatment, and this was using at day 45. But so that it's very interesting that this specific treatment tended to cause, again, more pathogenic bacteria outgrowth. Again, I want to mention the caveat that this is not the same as the human female reproductive tract microbiome. There's a lot of overlap, but it's not exactly the same. When we looked at clindamycin, so again, clindamycin is used to treat bacterial vaginosis, and what was interesting is what we saw was a tendency for bugs that are associated with bacterial vaginosis in humans, like Adipobium, Mobiluncus, Porphyromonas, Prevotella. We saw that there was decreases in these PV-associated bacteria with the clindamycin, so that is a good sign that oral clindamycin can have a potentially beneficial effect on these microbial communities. When we look at all, or the cephalexin and the clindamycin across the time points, then again we see that there's this kind of clustering, specifically at the 45-day time point, which is right in the middle of that antibiotic treatment. And sorry, I had two different students analyze this, so it's a little different nomenclature, but this would be our day 3 during antibiotic treatment is what that's equivalent to. So there's, again, this big shift very early after the treatment. But it does raise the question if the enrofloxacin and paramycin were less disruptive. So here we want to look at the average variance of the bacteria in each animal throughout time. And so the way we're doing this is the divergence across time points. So from baseline across time points longitudinally, and then this is each animal individually, and then it's colored here with the different antibiotic treatments. So on the far left, you have the enrofloxacin, again, each animal. So the higher up the bar is, the more divergence, the more changes that antibiotic induced. The paramycin is here, the cephalexin is here, and then the clindamycin, and this might just be easier to look at when we have the average of all the animals. And what you can see is that there's quite a bit of shift and divergence when you look at all of these different antibiotics, when you average animals together, except for with the enrofloxacin, again, not a bacteria, or not an antibiotic you might be as familiar with because this is mostly used in veterinary practice, but it was very interesting that that was the one, even though you still had shifts in immunity, it had the least disruption to the microbiome in the vagina. So just to wrap this part of the talk up, oral antibiotics disturb the stable vaginal microbiomes in healthy rhesusmic cats. The CD4 T cells increased and peaked for all groups very rapidly following antibiotic administration, and again, this could have implications for things like HIV transmission. You did see the CD4, CD8, and neutrophil spikes that coincided with significant increase in pathogenic taxa, particularly for the cephalexin-treated animals. And the enrofloxacin was significantly less disruptive to the vaginal microbiome. And I just want to point out that this is very, very new data, that a lot of these analyses were seriously done in the last couple of weeks. So I am very well aware we have a lot of work to do to follow this up and understand this better and understand what the affiliations are between and the correlations between the altered microbiome and the immunity here and what this potentially means for disease. But with that, I'd like to thank my group, Brian Chu and Andrew Gustin, did the majority of the work on the vaginal microbiome side. Alex Devin, a former postdoc in my group, is the one who originally ran the monkey study. The Washington National Primate Research Center, and particularly Jeremy Smedley, where we ran the monkey study, and then Mark Cameron, Cheryl Cameron at Case Western Reserve University, where they did the RNA-seq analysis, and then my many collaborators on microbiome that are always there to support and help us all understand as we're trying to understand the microbiome field. So thank you very much for your attention, and I'd be happy to take any questions.